Actinomyces turicensis, Schaalia turicensis, general information. Taxonomy of Actinomyces turicensis family. Actinomycetaceae, genus, Schaalia turicensis. Formerly, Actinomyces turicensis. Natural habitats. Actinomyces turicensis is a part of the normal microflora found in human mucous membranes. It can be isolated from clinical samples without necessarily indicating an infection. It is typically found in the vagina, gut, and skin, particularly in the lower part of the body. Clinical significance. Although usually harmless, Actinomyces turicensis has been associated with certain infections. It has been isolated from pilonidal sinuses, perianal and decubital abscesses, infections of the ear, nose, and throat, postoperative wounds, and intrauterine contraceptive devices, IUCDs. Studies suggest it may act as a potential pathogen, primarily in genital infections, followed by urinary tract infections and skin-related infections. Please note that while Actinomyces turicensis can be found in these infections, its exact role in causing them is still being investigated. Gram stain. Actinomyces turicensis are gram-positive, small, cocobacilli rods. It's important to note that they are not dipteroid, meaning their shape is irregular and not consistently club-shaped like some other bacteria. Culture characteristics. Actinomyces turicensis is a facultative anaerobic bacterium that grows in both air and CO2 environments. Colonies of Actinomyces turicensis on blood agar plates are gray, semi-translucent, smooth, low-convex, and have entire edges. These characteristics can lead to misidentification of Actinomyces turicensis as non-hemolytic streptococci, lactobacilli, or other commensal organisms. When grown on biolesculin agar, BBAO stroke, Actinomyces turicensis exhibits growth. Characteristics of Actinomyces turicensis. Gram-positive bacilli, meaning they retain the crystal violet stain in the gram staining protocol, appearing purple under a microscope. Medium bacilli, indicating their size falls within the medium range for rod-shaped bacteria. Growth both aerobic and anaerobic. Actinomyces turicensis can grow in the presence or absence of oxygen. Alpha hemolysis. Actinomyces turicensis cause partial breakdown of red blood cells, producing a greenish discoloration around their colonies on blood agar. Catalase negative. Actinomyces turicensis do not produce the enzyme catalase, which breaks down hydrogen peroxide. Oxidase negative. Actinomyces turicensis do not possess the enzyme cytochrome C oxidase, involved in cellular respiration. Urease negative. Actinomyces turicensis cannot hydrolyze urea into ammonia and carbon dioxide. Non-modal. Actinomyces turicensis lack the ability to move on their own. Vancomycin susceptible. Actinomyces turicensis are sensitive to the antibiotic vancomycin. Colistin resistant. Actinomyces turicensis are not affected by the antibiotic colistin. Cells irregular. The cells of Actinomyces turicensis are not uniform and can vary in shape. Poem for Actinomyces turicensis. In mucous realms, Actinomyces turicensis finds its home, a resident, benign, it seems, yet in infections, Actinomyces turicensis starts to roam, fulfilling pathogen streams. From sinuses to wounds, Actinomyces turicensis may arise, in ears and noses. Actinomyces turicensis finds its way, a potential foe in subtle disguise, in genital and urinary tracts. Actinomyces turicensis may sway. Gram-positive rods, irregular in form, on blood agar. Gray colonies unfold, a facultative anaerobe. Weathering the storm, in air or CO2. Actinomyces turicensis's story is told. Catalase negative, oxidase it lacks, urease it shuns, and motility it foregoes. Vancomycin's touch. Its spirit attacks, colistin's resistance. Actinomyces turicensis's resilience shows. Schaalia turicensis, once Actinomyces' own, in the microbial world. Actinomyces turicensis's presence is known.